We only see so far because we stand on the shoulders of giants. What do our ancestors have to teach us? Come with me on a journey to the past, here on Interview with a Dead Scientist. Hello, and welcome to Interview with the Dead Scientist. I'm your host, Christian Copley Salem, and I'm pleased to have with me today Gregor Mendel, the father of modern genetics. Welcome, Gregor.
Thank you. I'm happy to be here.
Tell us a little bit about yourself.
Thank you again for having me. My name is Gregor Mendel, and I am an Augustinian monk. I was born in Czechoslovakia, and as a child I loved to keep bees and worked a little as a gardener. As an adult, however, I'm just a monk who studies pea plants.
How did you get interested in pea plants?
Well, that is a long story. But basically, I joined the monastery to get a free education. It's so expensive nowadays. Anyway, I had some questions about the way traits were passed from one generation to the next. I started out looking at mice, but then the bishop came in, all in a flurry, and he told me straight out, "You can't study animal sex." So, naturally I switched to plants, particularly pea plants, because they have both male and female genitalia. Because of this, I could control their breeding by cutting out the male part. This way I could choose which plant to breed with which.
What did you discover by working with these pea plants?
Well, I found that some traits, like color of the flowers and such, came in two varieties, white and purple. When I crossed one plant with white flowers, together with one plant with purple flowers, I got all purple flowers in the offspring. As you can imagine, this was very disappointing. However, when I crossed their offspring, I was amazed because the white color returned in a small portion of them.
Mendel was unaware of just how important his discoveries would be. What he discovered when looking at the color of pea plant flowers was the concept of dominance. The different colors of pea plant flowers are called traits. Traits are detectable variants in some character of an organism. The eye color trait has many different variations: blue eyes, brown eyes, green eyes, etc. In the pea plant, the color of the flower is determined by variations in a single gene. One variation produces purple flowers, and another variation produces white flowers. These are called alleles. The gene for purple flowers is one allele of the flower color gene, and the gene that produces white flowers is another allele of that same gene. Like we saw last time when we looked at chromosome 15, the genes for eye color come in many alleles. So what we see is the trait, and the variation of those traits is caused by the underlying alleles. So when you were conceived, you received one set of chromosomes from each of your parents. This means that you have two alleles of every gene. Pea plants work the same way. When one of those alleles will override the other, it is called dominant. The weaker allele is called recessive. That means if you get a dominant allele from one parent and a recessive allele from the other, your expressed trait will match that of the dominant allele, even though you do have one copy of the recessive gene. This can help explain Mendel's purple flowers. Mendel started with an initial set of plants that bred true. This means that no matter how many times they were crossed with each other, they produced the same traits as the parents. If we looked at their genes, we would see that the purple plants had two alleles from the purple flowers, and the white-flowered plants had two alleles for the white flowers. We can designate them as P and W. This is the parent generation. So the purple flowering plants have two of the P alleles, like this, and the white flowering plants have two alleles of the W gene. We know from our study of meiosis that genes are sorted independently into the gamete cells. Because the purple flowering plants have two P alleles, all of their gametes will get a P allele. The same is for the white flowering plants. If we cross two plants, we are just mixing one gamete from each plant together to make a new plant with two N chromosomes. We can create a chart of the different possible allele combinations this would produce. On the top row, we can label the two possible alleles from the purple flowering plant, and on the side we can label the two possible alleles from the white flowering plant. Great. Now in each square, we can put what allele combination the offspring would get. So in box 1, we get one purple and one white allele, and in fact, we get that same combination in every box. The P allele here is dominant, And this explains Mendel's observation that when he crossed the white and purple plants, all he got was purple. All 4 possible combinations have one purple allele, and since it is dominant, that is the trait they will show, purple flowers. This is the F1 generation. F comes from philo, which is Latin for sun. Now is when things get interesting. Let's imagine what we get if we breed 2 of these offspring together. The children of 2 F1 generation plants would be the F2 generation, the second generation of children. We can visualize how the combinations of our F1 alleles would look. Here we put the alleles from an F1 plant on top, and then we put another one along the side. Now we can look at all the possible allele combinations that will be seen in the F2 generation. In the first box, we have 2 purple alleles, and in the second box, we have 1 purple and 1 white. We know that this will produce purple flowers as a trait. In the third square, we also have 1 purple and 1 white. Again, this will produce purple flowers. However, here in square 4, we will have 2 white alleles. This produces a white flowering plant. So the cross between the 2 traits gives us a 3-to-1 ratio of purple flowers to white flowers. This box is called a Punnett square. It's a way to help visualize the mathematics we will be discussing later. Let's kick this up a notch and look at 2 traits. Let's look at 2 alleles that have dominant and recessive variations. We can use flower color again, but this time we can add in the color of the peas themselves. Knowingly crossing 2 traits like this is called a dihybrid cross.
Can you explain your law of independent assortment? But of course. I discovered that different attributes of the plants seem to breed independently of the others. Plants with different colored flowers, or different colored pea pods, would mix as if each trait was selected at random. How this happens, I do not know.
The law of independent assortment that Mendel suggested has become an important concept in genetics. The modern version of this law states that alleles will be sorted into the gametes independent of each other. Remember that the chromosomes participated in crossing over during meiosis prophase 1. This allows alleles from homologous chromosomes to be sorted independently, so we can treat each allele as an independent trait. Starting with the F1 generation, we can make a Punnett square for a dihybrid cross. It's just a little bigger. Now we can list all the possible combinations of alleles that could be found in the gametes of each organism. Just as before, we will arrange all the possible gametes along the top for our first F1 organism, and do the same on the side for another F1 organism. We have the purple and white flower alleles, and we have the green and yellow pea alleles. Let's fill in our squares as we did before. What we find is that we have some purple flowered offspring with green peas, and some with yellow peas. Additionally, we have some white flowering offspring with green peas and also yellow peas. We have 9 purple flower and green pea plants. We then have 3 purple flower and yellow pea plants. Then we have 3 white flower green pea plants and only 1 white flower yellow pea plant. This is the ideal ratio for a dihybrid cross, 9 to 3 to 3 to 1. This works because the alleles for flower color and the alleles for pea color are sorted independently due to homologous recombination. Writing out a Punnett square and counting every box can be time-consuming. Let's take a look at these again from a mathematical perspective. So, if we look at the Punnett square for our single hybrid cross of flower color, we can make some intuitive statements. The first is that a white flowering plant will only occur in 25% of the offspring. That is 1 in 4. So there's a 1 in 4 probability that the offspring of this cross will have white flowers. On the flip side, there's a 75% chance or 3/4 chance that the flowers will be purple. What is the probability that we would get a purple flowered plant that carries the recessive white flower allele? It's 50%, or 2/4. Now we can ask a slightly more complicated question. What is the probability that the offspring would have at least 1 white flower allele? The answer is 75%, or 3/4. The mathematical way of doing this is by using the addition rule. The addition rule is used on single crosses when asking questions about the probability of one allele result and not another. So, the question what is the probability that the offspring will have at least one white flower allele, is really the summation of all the purple-white combinations as well as the all-white flower. To calculate this, we have to add the probabilities together. 2/4 for purple-white combinations and 1/4 for the all-white possibility. The addition rule is used when we are asking the probability of a particular state versus another. What happens when we add back in the genes for pea color? We get a much bigger square, and there are more probabilities we need to consider. We can ask questions like, what is the probability that the offspring will have purple flowers and yellow peas? To answer this, we can use the multiplication rule. This rule is used when two non-mutually exclusive events are happening at the same time. Because of the law of independent assortment, getting a purple flower allele and a yellow pea allele are independent. Thus, we are asking what is the probability of an offspring getting at least one pea allele and getting two recessive yellow pea alleles. We can mark them on the diagram. Mathematically, we can determine this by using the multiplication rule. First, we need to know the probability of getting at least one pea allele. We can look at the simplified Punnett square for this because remember that each trait is independent of the other. So what is the probability of getting two yellow alleles? It's 1/4. So if we multiply these together, we will get the probability that we have at least one pea allele and two yellow pea alleles. 3/4 times 1/4 is 3/16. Let's look at our large Punnett square and check to make sure we did it correctly. We can see that there are 3 out of the 16 squares that are both purple and yellow. That's fantastic. To summarize, in order to figure out the probabilities involving one gene, or this Punnett square, we use the addition rule. However, when we're asking about multiple genes, like in this Punnett square, we use the multiplication rule. What we've been looking at so far are simple cases. The purple flower color in our example displays complete dominance. This means the white and purple combination is just the purple flower. However, some genes show incomplete dominance. This means the traits can blend. Let's check out these other flowers that are red. The red flowers have incomplete dominance, which means if we breed them with white flowers, the F1 offspring will have pink flowers. Notice that when the offspring carry both the red and white alleles, they have pink flowers. This is incomplete dominance. Codominance is where both alleles are fully expressed as traits. An example of this is blood type. People who have one A allele and one B allele will have AB blood type. Thus, the traits don't blend or show dominance. They simply exist side by side. Both A and B alleles are codominant. Epistasis is another variation of the principles we have looked at. In epistasis, two genes interact in ways that produce a different ratio than the 9-3-3-1 we've already seen. In epistasis, one gene can rely on another gene to reveal its trait. A good example of this can be found in cats. Cats come in a variety of coat colors and patterns. However, there is a gene that can make a cat all white, and this overrides the other gene for color. If we look at a Punnett square for this type of breeding, we can place all the genes like we did before. This time, we are using the dominant white gene and the gene for coat color. In this case, the coat color is dominant brown, with the recessive allele being gray. Let's look at each possible offspring combination and see what this would look like. First, we see that any offspring with the dominant white gene is white, even if it has the dominant brown allele. This is because the dominant white gene is a gateway to any other color at all. If the dominant white gene is working, then no other color can be seen. However, in all of the non-dominant white offspring, we see either the brown dominant or gray recessive. This seems complicated, and it can be. The important thing to remember is that epistasis is when one gene regulates the expression of another.
Interesting. I had no idea my ideas would grow so much. Well, you have been dead for over 100 years, so indeed.
In people, scientists don't run around doing weird breeding experiments. Instead, we look at the results of freely chosen mates, and we draw some conclusions from what we find. This is called a pedigree analysis. A pedigree is a description of observed traits passed down through the generations. This can be an interesting exercise, but it can also be a valuable tool. One thing that we can use pedigrees to identify are recessively inherited disorders. Recessively inherited disorders are disorders that are caused by recessive genes. People who have the dominant allele would never experience the disorder, but they can be carriers. If two people who are carriers decided to procreate, they have a 1 in 4 chance of having a child with the recessive disorder. Many pedigree charts use symbols like these. Square is for a male, and circle is for female. The half-black square and circle are for carriers. Then the solid black circle and squares stand for people who have the disorder. That is to say, they have 2 recessive alleles. The word for this is homozygous. Homozygous means that both copies of an allele are the same. In this case, the disease is caused when a child is born homozygous for the recessive allele. Heterozygous means they are not the same. In the case of the recessive allele, it means that they carry 1 copy of the recessive and 1 copy of the dominant. Heterozygous means that the organism's 2 copies of an allele are different. If both of their alleles are the dominant allele, they are also homozygous, because homo means the same. Let's look at this couple's family tree and see how recessive disorders can stay hidden. Bill is a carrier of the recessive blood disorder. He does not have the disorder, but he is only a carrier. Mary is not a carrier and is homozygous for the dominant allele. Their children have no chance of having the blood disorder, because as we can see from the Punnett square, all of Bill and Mary's possible offspring carry the dominant allele. In this case, Bill and Mary have Charlie and Shayna. Both Charlie and Shayna carry the recessive gene, but neither have the disorder. Shayna marries Alex, who has 2 dominant alleles. Just like her parents, Shayla and Alex cannot have a child with the disorder, but they can produce carriers. However, Shayla's brother, Charlie, has a child with Nina. Both Charlie and Nina are carriers. They have a 1 in 4 chance of having a child with the blood disorder. Their child, DeAndre, is born with the blood disorder because he has 2 recessive alleles. It's easy to see how such conditions can sneak around within a family tree without showing up until much later on, even if none of the recent ancestors had the disorder.
It's truly fascinating how complex and interesting genes are. I guess I only just scratched the surface. That's true, but your work was very important, and I want to thank you for being here today. Thank you. Thank you very much for having me. From all of us here at Interview of the Dead Scientist, we'd like to wish you a good night, and thanks for watching. Is there any way to get a burger around here?